So moving on to the RNA viruses. So we will cover the enveloped and non-enveloped RNA viral families. So all of these are single-stranded as far as the RNA genome. The only exception here is the Reoviridae. So we'll talk about the Orthomyxo, Bunya, Arena, Paramyxo, Rhabdo, Philo, Toga, Flavi, Corona, the Retroviridae, PicoRNA, Calisi, and the Rio, which again are the only dsRNA genome out of all of these. So the Orthomyxo, this is the fancy viral family name for influenza or the flu. So this is a single-stranded RNA virus, and there are two types here, influenza A and B. The difference being that A circulates throughout many species and B is unique to human beings. Now, I would like you guys to remember two methods for how influenza viruses actually can mutate. So these two types of changes are referred to as antigenic drift versus antigenic shift. And the way to remember this is to think of these specific parts of any garden variety influenza virus. So whether we're talking about influenza A or B, each influenza virus has an H and an N that has been characterized. So I'm sure we've all heard of the H1N1, the swine flu, right? So H stands for hemagglutinin, N for neuraminidase. Neuraminidase is an enzyme that's involved in the fusion and entry process during viral replication, and hemagglutinin is the spike, the glycoprotein spike. So these two seem to be the best indicators for any mutagenic changes. So in the case of antigenic drift, this is a subtle minor variation of one of these components. So in antigenic drift, the hemagglutinin may change very subtly. So you still have the H1, but it may be structurally different based on one amino acid. Whereas in the case of shift, you have a completely new H, and this can be the result of homologous recombination or genetic recombination, whereby if you are infected, let's say, with two different types of flu virus, so H1N1 and H2N7, those two viruses could recombine to produce a novel virus with a new spike. So this is why every year there are new flu vaccines that have to be synthesized due to antigenic drift and shift. So as far as the flu, influenza type A, it's an acute, highly contagious respiratory illness. It's seasonal and it's associated with pandemics. It is among the top 10 causes of death in the US, and it is transmitted via respiratory droplets. So the virus will bind to ciliated cells of the respiratory mucosa, and it causes rapid shedding of the cells, stripping the respiratory epithelium, and result in severe inflammation. So we all know the common symptoms of the flu, right? Fever, headache, myalgia, a sore throat, shortness of breath, and coughing. And we do have antiviral meds, Tamiflu, that can be used. And as we know, there are vaccines that are synthesized every year. So as far as the types of vaccines, there's the shot or the nasal spray. So for the flu shot, this is an inactivated vaccine containing killed virus. So this is basically synthesized in such a way that the virus is grown using chicken cells or host cells and then heat or chemical inactivated. So it does not have the potential to revert back to the wild type virulent form. This one's usually given with a needle, usually in the arm, versus the nasal spray, which this contains live attenuated viruses. So these are viruses that again have been cultured in the lab using host cells, and they have been passaged through many cycles of replication until they are attenuated or changed in such a way that they would not cause the full-blown variety of the flu. So it will induce an immune response, but it won't actually make you sick. So this is sometimes called LAIV for live attenuated influenza vaccine, or FluMist. And for the vaccines, each vaccine contains 3 influenza viruses, 1A, H3N2, H1N1, and then 1B. And then they change every year according to what is actually circulating. So as far as influenza is concerned, the one that really we should be concerned with here is the bird flu. So this would be the H5N1. Now this is the result of antigenic shift, which causes new combinations of proteins on the surface, so those new spikes. And if this new or novel virus spreads easily from person to person, this worldwide epidemic can occur, referred to as a pandemic. If we don't have immunity, we haven't seen this before, especially if we don't have a vaccine, this could cause some serious issues. So this H5N1 seems to have genetic information or genetic material from pigs, birds, and humans. And so this new strain that we have been seeing circulating, and this has only been from birds to humans, could cause disastrous effects. Now, the only reason why we haven't seen a pandemic is because of the fact that it is only transmissible from bird to human and not from human to human. But when that happens, watch out. So this is one to keep an eye out and keep your ears open as far as any changes that are occurring throughout the world with how this virus actually evolves. So on to the paramyxo. So these include the parainfluenza, mumps, measles, and RSV. So these are all transmitted via respiratory droplets. And these viruses cause the infected cells to fuse with neighboring cells. So this cytopathic effect is referred to as syncytium, or also multinucleated cells can form. So these are cells that have multiple nuclei. And this is an electron micrograph showing a swollen multinucleated cell here on the right, this syncytia phenomenon that I was just referring to. So this would be the fusion of cells, and this actually allows the virus to replicate more efficiently. Now, starting off with parainfluenza, it's as widespread as influenza, but it is generally more benign, transmitted via respiratory droplets, and most commonly seen in children. And all respiratory symptoms, so minor cold, bronchitis, even later stages pneumonia, and barky cough is one of the associated resultant symptoms with a hoarse voice and some breathing difficulty. No specific treatment or vaccine seems to be available. Now, with regards to mumps, this is also known as epidemic parotitis or infection of the parotid gland. So this would be the salivary glands. And humans are the only reservoir. There's actually now only about 300 cases in the US per year, thanks to the advent of the MMR vaccine. So this is a live attenuated vaccine that has protection against measles, mumps, and rubella. The incubation for this virus is around 2 to 3 weeks with resultant fever, muscle pain, malaise, and this classic swelling of both cheeks because of the effects on the parotid gland. Now, it seems that there are complications here in 20 to 30% of infected young adult males. Epididymis and the testes can become infected, and sterilization can occur, but it is rare. So this is that characteristic swelling of the cheeks in the case of mumps. And then on to measles. So there's a big difference between the virus that causes measles and German measles, right? So for measles, this is caused by the morbillivirus. It's also known as red measles and rubeola. Very contagious. Again, that MMR contains protection against the morbillivirus. It's transmitted via respiratory aerosols. Now we're seeing less than 100 cases per year, but this has since gone up with the wave of the anti-vax movement. And in this case, the morbillivirus will invade the respiratory tract, causing sore throat, cough, headache, conjunctivitis, swollen lymph nodes, fever. And one characteristic symptom to remember here are these oral lesions known as Koplik spots. So these form within the mouth, and also this associated rash. So we see a rash with German measles as well, but not the Koplik spots. So just comparing these two. So I would remember rubeola, causative agent of measles, and this is the morbillivirus. And for this particular version of measles, again, the oral lesions are present, and we'll talk about rubella in a minute. So again, here are these Koplik spots, these oral lesions, and this characteristic rash. Okay, so we have the Koplik spots, and usually the rash, a red rash on the head, neck, or trunk. Now, there is a serious complication associated with the morbillivirus. It's rare, 1 case in a million infections, but it can lead to this SSPE. So this is subacute sclerosing panencephalitis. This is a progressive neurological degeneration of the cerebral cortex, white matter, and brainstem. And it involves a defective virus spreading through the brain by cell fusion, and it can destroy cells as a result, leading to coma and death in months or years. And again, this is provided in that MMR vaccine. And here's a cartoon image of the morbillivirus. On to RSV. So RSV stands for respiratory syncytial virus, also called pneumonia virus. And this will infect the upper respiratory tract and produce giant multinucleated cells. It is the most prevalent cause of respiratory infection in children 6 months or younger. And the very young are most susceptible to serious disease. So it will infect the epithelial cells of the nose and eye as the portal of entry, and it will then replicate in the nose and throat, causing rhinitis, wheezing, even ear infections, croup, and treatment seems to be, what is effective here is this monoclonal antibody that will act to actually block attachment to the epithelial cells of the nose and eye. And this drug is referred to as ribavirin. So on to rabies. So there's only 1 or 2 human cases per year. And remember, this virus is shaped like a bullet. So it also has an envelope, and it causes a slow progressive zoonotic disease. So the rabies virus will enter through a bite wound, grow at the trauma site for a week, and then enter the nerve endings and advance towards the spinal cord and the brain. So as far as treatment, it must be passive and active post-exposure immunization before it reaches the spinal cord and brain. Otherwise, it is fatal. And as far as symptoms, similar to what you see in rabid animals, agitation, disorientation, seizures, twitching, foaming at the mouth, also paralysis and stuporous behavior. So around here we see raccoons being at the top of the list for spreading the virus or harboring it. Also skunk and fox and even bats can spread the virus. And then on to the coronaviridae. So these, if you look at the electron micrograph, have distinctively spaced spikes on their envelopes, very common in domesticated animals, and there's 3 types worthy to mention here. So HCV causes a cold. There's also an enteric virus. And then the most severe of this family would be SARS. So this stands for Severe Acute Respiratory Syndrome. And it's thought that the origin of SARS was from a civet cat, which is consumed as a meal in certain parts of the world. So China, it seems that this animal may have actually started the SARS outbreak. So moving on to rubella, which is caused by a toga virus. So it's the ruby virus. And this is another common name, German measles for rubella. So remember, we already brought up measles, which was caused by the morbillivirus. And I mentioned that the Koplik spots in the rash were characteristic of that disease. Here we still see a rash, but remember that for rubella, there is a concern here for congenital defects. So this virus is known as a teratogen, meaning that it can cause birth defects. And usually if a female becomes infected within the first trimester of the pregnancy, a miscarriage will occur. If it's past that, then you have these congenital defects. So this is transmitted through contact with respiratory secretions, and the MMR vaccine does provide protection. So again, in the case of congenital rubella, infection during the first trimester will most likely induce miscarriage, or if it's after that, multiple defects such as cardiac abnormalities, ocular lesions, deafness, and also potentially mental and physical retardation. And then there's postnatal rubella, which is associated with malaise, fever, sore throat, swollen lymph glands, rash, and this is more generally mild. So moving on to the arboviruses, so these are all spread via insects. So arthropods, and there's more than 400. A lot of these are actually at the top of the list for newly emerging infectious diseases, and they fall into many different viral families, so Toga, Flavi, Bunya, and Rio. Most illnesses associated with the arthropod spread arboviruses are mild fevers, but some can result in severe encephalitis. So dengue fever, Western and Eastern equine encephalitis, and also yellow fever are some of those that could cause more severe disease. So we see the West Nile virus around here, but these other ones, the yellow fever and dengue fever, there is talk of them making their way to the continental US because of the increase in the population of the insect that's actually spreading the disease. But generally, we see Colorado tick fever and Western equine encephalitis and West Nile virus as well. Also, Eastern equine encephalitis around this area. So as far as the Togaviridae, so some of these viruses can cause either dengue fever, yellow fever, or West Nile. So we've seen it here in Connecticut even, but very rare cases. And the cases that have been reported, usually they're in much older individuals, so people who are immunocompromised. And the encephalitis does not occur in every case. Now, with dengue and yellow fever, more commonly, these can be fatal. So dengue, also known as breakbone fever, and it causes severe pain in the muscles and joints. Infections usually are mild, but it can be lethal. And as I mentioned before, there is concern over the possible spread to the continental US because of the increase in the insect population. So that would be the mosquito, and also yellow fever. So similar symptoms here. This may progress to organ failure and death. Now, as far as dengue, the distribution here of the disease is reflective of the distribution of the insect that's spreading it. So we can see that the populations have increased over time, and there's worry that it will spread to the US. Now, if you look, this is the particular genus species of the mosquito that actually spreads it. So I would like you guys to remember this, the Aedes aegypti mosquito. Now, another virus that is spread via the mosquito is the West Nile virus, and we have seen indeed cases here in Connecticut. It's estimated that about 20% of people who become infected will develop West Nile fever. So the symptoms include fever, headache, fatigue, body aches, occasionally with a skin rash, swollen lymph glands, and the illness can be as short as a few days, but even healthy people have reported being sick for several weeks. And about 1 out of 150 infected people will develop severe disease, so encephalitis and meningitis. And the incubation period is anywhere from 2 days to 2 weeks. Now, just to note here as far as the transmission cycle, so it does cycle through incidental hosts, so that would be humans or other larger animals, and even birds can serve as a reservoir host for the virus. On to picoRNA viruses. This literally translates to small RNA. Hep A is one that falls within this family. It causes the most acute form of hepatitis, and this is characterized by fecal-oral transmission route. There is an inactivated viral vaccine called Havrax, but this one, typically we don't see any long-term sequelae or cancer long-term effects from it. So it's more of an acute disease. And then rhinovirus, there's over 110 different serotypes. This is the most common cause of the common cold. And then we have polio, which is associated with the most severe disease out of all three of these. So polio is a pretty hardy virus. It's resistant to acid, bile, detergents, and also can survive stomach acids. And it is contracted by ingesting it through food, water, or contaminated objects. Most infections are mild. So as far as the pathology, it's ingested, it will grow in the oropharynx and intestine, and then can spread to the blood. So in this case, that would result in viremia. So the blood contains a high amount of virus. It can spread to the spinal cord and brain and lead to paralysis. And there are cases of PPS, or post-polio syndrome, which can result in muscle deterioration. So this is electron micrograph, cartoon image. And because of these potential long-term effects, a vaccine was developed. It was an inactivated form. Also, there was an attenuated form that actually was showing reversion back to the wild type. But this particular pathogen, we've seen no natural cases of polio since 1991. And at the height of the epidemic in 1952, nearly 60,000 cases were noted with more than 3,000 deaths. And that was just in the United States alone. And also, as a sidebar, if anyone saw the recent 60 Minutes special documentary about a group of medical doctors using a genetically modified form of polio and injecting it into glioblastoma tumor. So glioblastoma is the most malignant form of brain cancer. Usually people who are diagnosed die within 3 months. And they've actually found that with this genetically altered version of the virus, that it will kill these particular cancer cells, and they've even seen in some cases that it's cured people. So I know it's a progressive, innovative, kind of very new treatment, but it seems to hold some promise for treating that highly invasive brain cancer. Okay, moving on to the Rio viruses. So these are non-enveloped, segmented, and these are DS. So the Rio itself is not a significant human pathogen, may cause upper respiratory disease or the common cold, similar to the common cold. And then rotavirus within this family, more clinically significant, globally causes 50% of cases of diarrhea and death of over 600,000 children. So this is a big one. And this is just showing you the geographic distribution of the rotaviral cases. All right, so Ebola is one of my favorite viruses out of all of the viruses we talked about this semester. So it's one of the most controversial because it is high on the list as far as a potential bioterror weapon. And it's a scary thought to think about if this virus were to be aerosolized, because there's no current approved treatment. There may be a vaccine on the horizon. The only reason being that in the last year, if anyone kept up with the international news, that Ebola seemed to be spreading in certain West African countries, Liberia, Sierra Leone, Nigeria, and it essentially killed half of the people that it infected. So this included a lot of medical missionaries and doctors from the United States. And for the first time, Ebola actually made its way out of West Africa and came to the US. So Dallas, Texas, Thomas Duncan was infected in Western Africa and then traveled on a plane and brought it to the US and infected one of the nurses that was caring for him. So it's a very scary illness because it is fast acting, and again, we don't have the greatest treatment available. So Ebola is a virus caused disease limited, we can change this, to parts of Africa. So this last year, again, we saw it here in the US. When you are infected, within a week, you will develop a fever, a raised rash, internal bleeding, and the bleeding from the mucous membranes is typical, causing bleeding from almost every orifice, so the mouth, nose, eyes, and the rectum, and it seems to be transmitted through bodily fluids, but it is a very hardy virus outside of the human body. Also is shed in tears and on the surface of the skin. So there are a lot of controversial books written about this particular pathogen. Again, it could be used as a bioterror weapon, so a very scary thought. So a virus that we do see here in the US that has a 50% mortality rate is the hantavirus. So this is found actually worldwide, and it is spread, it has this unique transmission route. It's spread via contact with rodent waste or urine. So this is a very dangerous respiratory illness leading to hantavirus pulmonary syndrome, and we don't really have an effective treatment. Again, 1 out of every 2 people will not survive, so this is another one to be concerned with. And then we have the retroviruses. So the one that I would like you guys to really familiarize yourselves with is HIV. So this particular virus has within its genome this enzyme called reverse transcriptase, which will take a copy of their RNA genome and convert it to DNA. That DNA can then be integrated into the host cell's chromosome. So it actually becomes integrated into the chromosome of your CD4 T helper cell, and then that becomes what we call a provirus or a memory cell. So HIV stands for human immunodeficiency virus. It is the cause of, in its tertiary or later stage, AIDS, which is acquired immunodeficiency syndrome. And also within this group here are the T cell lymphotrophic viruses, which can cause leukemia. So specifically for HIV, it still seems to be a major problem internationally, and I do believe that we will have an appropriate treatment or cure for it in the near future, but we're not there yet. And there's multiple reasons for that, one of which is that it's highly mutagenic, so it has a very poor proofreading capacity, meaning it seems to be mutating at a high rate. So very frequently as it's replicating, it will adapt to whatever drug is thrown at it. So we saw it first emerge in the early 1980s. HIV 1 and 2 subtypes are not closely related, but it seems like they may have originated from a very closely related virus called SIV, which is simian immunodeficiency virus. So this is found in chimps. 1959 was actually the first documented case of it, and it's found in blood in the highest titer, semen, and vaginal secretions. So high risk choices here or activities would be sex, sharing needles, and mother to child. And it does not survive long outside of the body. So the specific target cell here are the T helper, the CD4 T helper cells, and macrophages. And the first signs of AIDS are opportunistic infections such as pneumocystis, so this is caused by a fungus that would not cause disease in a healthy individual, and also HHV8. This is a herpes virus that can lead to, it's similar to melanoma or skin cancer, and in late stage HIV patients it's called Kaposi sarcoma. So this is actually a picture of it. And by the tertiary stage of disease, so this is as it's progressed, so following infection up to 10 to 15 years post the time of infection, basically since the virus is targeting the immune system, by that late stage, 10 to 15 years, HIV-infected patients basically have no immune system left. So they are just riddled with all of these opportunistic infections. Everything from protozoal viral, fungal, and bacterial diseases, and also to cancer. So everything from toxoplasmosis to the Kaposi sarcomas I just showed you, caused by a persistent herpes infection. Also, these persistent diarrheal conditions, wasting syndrome, persistent fever, and a ton of viral, fungal, and bacterial. So CMV, Epstein-Barr, herpes zoster, pneumocystis is high on the list. Also tuberculosis, salmonella, even pelvic inflammatory disease. So basically, these individuals have a very weakened immune system. It's almost completely wiped out. So homosexuals or bisexual males account for 45% of the cases. Also, intravenous drug users, 30%. Heterosexual partners of HIV, around 11%. And blood transfusions and blood products. So since the 80s, this does not pose a serious risk. So it's usually for people who are abusing drugs with the use of needles that will contract it, not actually receiving a blood transfusion in a hospital. There were a lot of deaths as a result of this in the 80s, and now there's this whole screening process to ensure that blood is not contaminated with any pathogens. Also 9% inapparent or unknown risk. And there's congenital or neonatal, accounting for 5%. Medical and dental personnel, 1 in 500 chance of contracting it from a needle stick accident. So this is just kind of summing up what I just showed you about high risk activities. And then here is step-by-step the viral replication cycle. So HIV has these glycoprotein spikes on the surface called GP120 or 160. They will bind to the CCR5 receptor on the CD4 T helper cell. It will then fuse with the cell membrane. It has an envelope. So following this fusion, reverse transcriptase, which is one of those enzymes that it carries with it in its genome, will take the RNA and convert it to DNA. That DNA then gets integrated into the host chromosome, and we can then call that cell a provirus or a memory cell. It can produce a lytic infection or remain latent. So there's kind of 2 cycles that it can take. So this is a cartoon image here. Main components that you should remember are the spikes, its RNA genome, also has an envelope. Reverse transcriptase, integrase, and protease are important enzymes that are necessary for its replication cycle. So there are 3 stages. In the initial stage, usually people don't have symptoms. Secondary stage is when you see a lot of common cases of oral thrush, so persistent yeast infections of the mouth. And then that last stage is when you see the T4 cell levels fall to a ridiculously low amount, and this is when you start to see all of those opportunistic infections. So as far as early on, antibodies are actually detectable 8 to 16 weeks after infection, and then it may progress into that latent stage. So the latent stage would be when the reverse transcriptase actually carries out its enzymatic activity, converting RNA to DNA, and then the integrase integrates the genetic material into the host cell. So how do we treat it? We'll stop every step of the way. So inhibit these enzymes, stop fusion or entry, inhibit the translation. So remember, every virus needs to replicate its genome and also produce all of the necessary proteins. There's no current vaccine. As far as prevention, avoid all of those high-risk activities I mentioned. And so that concludes the RNA viral chapter, chapter 25. And this also concludes the YouTube lecture series. So I hope you all enjoyed this class online, and I wish you all the best of luck for the last exam, Lab Practical 2, and also the final. And best of luck to all of you in your future endeavors. Take care.